Hello everyone, welcome all of you in this hematological section of human physiology. Today I am going to talk about hemoglobin. What is hemoglobin? Hemoglobin is derived from two words, Greek hema that is blood and Latin globus that is ball, blood ball. And it is present in hemoglobin is present in RBC, red blood cells, and due to presence of this hemoglobin, these cells RBC are red. And this is an iron containing metalloprotein in RBC. Iron containing metalloprotein in RBC, red blood cells or erythrocytes. Two things are there, globin, polypeptide globin attached with a heme pigment. Synthesis. Synthesis occurs in mitochondria and cytoplasm of proerythroblast, early normoblast, intermediate normoblast and late normoblast. In mitochondria, succinylcholine and glycine obtained from metabolites are used to form pyrrole ring. Pyrrole rings are basic structures of hemoglobin. When four pyrrole rings combine, they form tetrapyrrole ring. Tetrapyrrole ring is also known as protoporphyrin 9. Tetrapyrrole rings. Pyrrole ring is the basic unit and four will form tetrapyrrole ring. This is also known as protoporphyrin 9. Protoporphyrin 9. Tetrapyrrole rings are joined together by methane bridges. Pyrrole rings are numbered 1, 2, 3, 4 according to their position, and the carbon atoms of this methane bridges are named alpha, beta, gamma and delta. The carbon of pyrrole rings is given 1 to 8 numbers on which side chains are attached, 1, 3, 5, 7, and methyl group 1, 3, 5, 7 combines with methyl group, 2 and 4 with vinyl group, 6 and 7 with propionic acid. Iron is in ferrous form, binds with the nitrogen of each pyrrole rings. Iron in ferrous form binds with the nitrogen of each pyrrole rings. Ferrous iron in the mitochondria to form one heme molecules. Iron in the center of porphyrin and coordinates with the four nitrogen atoms in the center of the pyrrole ring. Now this forms a heme. This forms an heme. One hemoglobin heme. Heme is covalently bound to the globular protein, globin, via the nitrogen atoms of the imidazole group below the porphyrin ring. Now this joins with this globular protein. Globular protein or globin is protein formed by four polypeptide chains. Polypeptide chains are synthesized in the cytoplasm. Four types of polypeptide chains are there. One pair of two polypeptide chains form the globin. Four types of polypeptide chains. Alpha chains 141 amino acids. Alpha chain contains 141 amino acids. Beta chains contain 146 amino acids. Gamma chains also contains 146 amino acids, but position of 37 amino acids differ from that of beta chains. Delta chains also contains 146 amino acids like that of beta chains, but 10 amino acids position is different. This form and this globin is formed. Now heme is attached. Heme is covalently bound to the globular portion, globular protein, that is via the nitrogen atoms of the imidazole group below the porphyrin ring. Now one hemoglobin molecule is formed. And one ferrous molecules bind with one oxygen molecule, that is two atoms. Hemoglobin 4, that is 4 tetraparolines. Therefore, there is 4 oxygen molecule or 8 atoms of oxygen molecules. It may be written as Hb4O8. And 1 gram of hemoglobin contains 0.3 milligram ferrous iron. 1 gram hemoglobin contains 0.34 milligram of ferrous iron. What are the substances essential for this hemoglobin synthesis? Protein is the basic. Then metals, iron, copper, cobalt, nickel, zinc, calcium. These are trace elements but very essential for this iron synthesis and hemoglobin synthesis. Vitamin B6, vitamin C and folic acids are also essential for development and maturation of this RBC, hemoglobin synthesis. These are the steps of synthesis of hemoglobin. Types of hemoglobin. In adult, types of hemoglobin is known as HbA, that is 98%. 98% of adult hemoglobin is hemoglobin A, which contains 2 alpha and 2 beta chains. And alpha chains contains 141 amino acids, beta chains contain 146 amino acids. Then HbA2, 2 alpha and 2 delta chains. HbA2 is also present in normal adult, but it is very little amount, 2% is there. And hemoglobin F is present in fetus and early childhood. 2 alpha and 2 gamma chains are present in hemoglobin F in children. And one thing is glycosylated hemoglobin. HbA1c is important because it is examined for diabetes mellitus. In diabetes mellitus, HbA1c glycosylated hemoglobin is estimated. Here, glucose binds with terminal valine in each beta chains. Glucose binds with terminal valine in each beta chains of hemoglobin A by a covalent bond. Beta chains are present in hemoglobin A, therefore it is only in HbA. HbA by a covalent bond. 3 to 6% is average in healthy person. Blood glucose level for 120 days. This shows that blood glucose levels for 128 days was within normal range. When it is 3 to 6%. When it rises, that means blood sugar control is not proper. This is in earlier stages, in embryos. In embryos, there are other two types of normal hemoglobin, Grover 1 and Grover 2, and hemoglobin Portland. Grover 1 is 2 zeta and 2 epsilon. Grover 1 contains 2 zeta and 2 epsilon chains. Grover 2 contains 2 alpha and 2 epsilon chains. Hemoglobin Portland 1, 2 zeta and 2 gamma, and hemoglobin 2 is 2 zeta and 2 beta chains are present in Grover. This is about Grover 1 and 1, 2 and hemoglobin Portland 1 and 2. These hemoglobins are present in embryo stage. Now, pathological hemoglobin. These are physiological hemoglobin. What are physiological hemoglobin? HbA, HbA2, HbF. And in early stage, Grover 1 and 2 and hemoglobin Portland 1 and 2 are present. What are the pathological hemoglobins? There are many pathological units, but important pathological hemoglobins are hemoglobin S, AS, hemoglobin C, E, Hopkins, D chain, D, and others. Hemoglobin H, that is hemoglobin H 4 beta chains. Hemoglobin H contains 4 beta chains. There is no alpha chain. And hemoglobin B contains 4 gamma chains, no alpha and no beta chain. These are the pathological hemoglobins. Pathological hemoglobins, HbS present in sickle cell, HbAs, HbC, HbE, Hopkins D, etc. HbH, for special mention, they contains no alpha chain, only beta chain, 4 beta chain. But hemoglobin BARTs, that is 4 gamma chains are present. These are the abnormal hemoglobin. So in this, we learnt about what is hemoglobin, synthesis of hemoglobin, where it is synthesized, and what are the essential factors essential for this formation of hemoglobin. Then types of globin, types of normal hemoglobin and pathological hemoglobin. In next class, in next lecture, I will talk about the functions of hemoglobin and fate of hemoglobin. Function and fate of hemoglobin in some detail. Okay, thank you for watching. You may share, please share it and follow the channel. Thank you.